mushrooms seem to have conversations with each other, especially after rain. Mushrooms can talk to each other. Such conclusions were reached by Japanese scientists in a recent study. According to them, rain may induce some of them to communicate using electrical signals. Some fungi play a key role in maintaining trees in the forest. One such example is ectomycorrhizal fungi, commonly found on pines, oaks and birches. They form a sheath around the roots of trees and develop into vast underground networks that draw essential nutrients from the soil to feed themselves and other plants in this symbiotic relationship. This is an example of ectotrophic mycorrhiza, also known as external mycorrhiza. This network also seems to be used for communication between fungi. For a new study, researchers at Tohoku University in Japan conducted field tests on mushrooms and found that their electrical activity increases after rain, suggesting that these mushrooms become talkative in response to rainfall. The description and results of the research were published in the journal Fungal Ecology. It will probably seem surprising to many people, but mushrooms growing in the forest may be able to talk to each other. Scientists from Japan undertook a more thorough study of this issue in the local mixed forests, although, of course, not only there, in Poland as well, mushrooms called Laccaria bicolor grow. It is a fungus quite useful for trees with which it lives in symbiosis. Even for oaks and pines, it helps them to supply water and nutrients, receiving carbohydrates in return. It is even believed that these fungi may contribute to the fertilization of trees. And they do so in a rather ruthless way. They use small insects called springtails, which they first lure and then kill with toxins. Of course, both trees and mushrooms benefit from this by consuming nitrogen obtained from the insect. However, in order for the symbiosis of two-colored lacewings and trees to be possible, the existence of appropriate connections between them is necessary. And this is indeed the case. These fungi build a kind of covers around the roots of trees, nets with which they entwine them. They do not penetrate directly into the tree, and their hyphae form extensive underground networks. But what is this understanding about? Well, previous research has shown that mushrooms have a certain potential to generate an electrical charge that activates in response to changes in the surrounding environment. And already in this context, it was speculated that this is how fungi can communicate with each other. For example, during research from 2022, it was found that this creates electrical activity in the style of nerves. It was even possible to isolate about 50 words spoken in this way by mushrooms. This time, however, the scientists decided to study the communication between the fungi under controlled, albeit still natural, conditions. Finding a group of lacewings growing near a sawtooth oak, they decided to attach electrodes to them to monitor their electrical activity. One day each was devoted to this in late September and early October. And while that first day was sunny and nothing noteworthy in terms of conversations of mushrooms, in early October a typhoon passed over Japan, bringing heavy rainfall. And then it started. The electrical activity of the mushrooms has clearly woke up. In the beginning, the mushrooms showed a lower electrical potential and we attributed this to the lack of precipitation. However, after the rain, the electrical potential began to fluctuate, sometimes exceeding 100 millivolts, said Yu Fukasawa, the study's lead author. At the same time, it was possible to notice that the level of this activity was dependent not only on humidity, but also on temperature. In addition, The strongest signals were recorded between individuals growing closest to each other. Traces of an ancient culture dating back 40,000 years have been discovered in China years. During archaeological excavations in northern China, researchers came across a unique set of tools and traces of the use and processing of ochre, a pigment extracted from rocks and clays. Based on radiocarbon dating and other analyses, they determined that habitats of hunter-gatherer communities were present in these areas as early as 40,000 years ago. Describing their findings, the researchers said the artifacts provided new information about the spread of human culture and technology around the world as Homo sapiens populations spread out of Africa. Archaeologists found traces of an ancient culture in northern China at the Xiamen Bay site, about 160 kilometers west of Beijing. The place is famous for its rich Paleolithic heritage. About 2.5 meters underground, scholars have found a veritable treasury of artifacts, numerous animal bones, remains of a hearth, tools made of stone, and evidence of the use and processing of ochre. Especially the latter attracted the attention of scientists. They show that the ancient inhabitants of these areas may have been the first people in East Asia to use ochre. Published in the journal Nature, research conducted by an international team of scientists shows the life of hunter-gatherers in what is now China, 40,000 years ago. While previous studies have shown that Homo sapiens arrived in northern Asia around 40,000 years ago, much about the lives and cultural adaptations of these communities and their possible interactions with archaic groups remains unknown. The archaeological site of Xiamen Bay, with its rich history, provides one of the best opportunities to understand the evolution of cultural change in Northeast Asia. Xiamen Bay stands out from other well-known archaeological sites in China because it has an unknown set of cultural features, said Dr. Feigang Wang of the Hebei Provincial Institute of Cultural Relics and Archaeology, whose team first conducted the excavation. One of these features is the extensive use of ochre, as evidenced by artifacts used to process large amounts of pigment. Within the band of sediments dated to 39 to 41,000 years ago, scientists found several pieces of ochre with different mineral compositions, suggesting that they may have been brought here for processing. The researchers also found an elongated slab of limestone with smoothed areas and ochre stains. The analysis carried out by scientists from the University of Bordeaux, led by Professor Francesco Derrico, points out that various types of ochre were brought to Xiamen Bay and processed by churning and grinding to produce powders of various colors and textures. As the researchers point out in the publication, the finds do not allow to determine the purpose of ochre processing. Perhaps it was processed to dye various objects or animal skins. Perhaps it was used for ritual purposes. However, the amount of ochre powder produced was large enough to permanently stain the surroundings of the place where it was made. This is the earliest known case of ochre processing in East Asia, the researchers emphasize. The researchers also found 382 tools. They are different from those found in local excavations. As they indicate, they were produced using two different techniques. In the publication, the researchers describe them as multifunctional and miniature. More than half of them are less than 2 centimeters. Some of them were also attached to the handles. Analysis of the remains found on the tools suggests that they were used for drilling, planing, cutting animal flesh and scraping animal skins. Ancient craftsmen made these multifunctional tools. demonstrating a complicated technical system for processing raw materials, unparalleled in the surrounding sites, which gives Ziema Bay an original character, the scientists explain. The ability of hominins to live in northern latitudes, with cold and highly seasonal environments, was likely facilitated by cultural evolution in economic, social and symbolic adaptations, said Dr. Shixia Yang of the Chinese Academy of Sciences and the Max Planck Institute for the Science of Human History in Germany. The Ziema Bay finds help us understand these adaptations and their potential role in human migration. So far, no human remains have been found at the Ziema Bay site. But the presence of Homo sapiens fossils has been found in nearby archaeological sites, leading scientists to conclude that this ancient workshop was the work of Homo sapiens. The authors of the publication remind us that modern humans are believed to have arrived in the region around 40,000 years ago. Although the nature of their interactions with the local inhabitants, Neanderthals or Denisovans, remains a mystery. Current evolutionary models suggest that Homo sapiens spread rapidly across Eurasia in a single wave, although the study authors admit that this theory seems overly simplistic. Varied technology and the presence of some innovations, such as small, precise stone tools and ochre work, but the absence of others, such as tools made of bone or bone ornaments, may reflect an early attempt by modern humans to colonize the area. This period may have involved genetic and cultural exchange with the aforementioned archaic groups, possibly replaced by successive waves of migration. Scientists have developed tomatoes that produce vitamin D using CRISPR-Cas9 gene editing technology. Scientists have created tomatoes rich in vitamin D. This vitamin is essential for the proper functioning of the body, and genetically edited tomatoes can help millions of people with its deficiency. New research shows that with a little help from gene editing, tomatoes can stock up on the precursor of vitamin D, an essential nutrient normally found mostly in animal products. The researchers used gene editing to turn off a specific molecule in the plant's genome, which increased provitamin D3 levels in both the tomato fruit and leaves. The action of sunlight, exposure to UVB light, then converts provitamin D3 into the active form of the vitamin. The results and description of the research were published in the journal, Nature Plants. Vitamin D is made in our bodies after exposure of the skin to UVB radiation, but its main source is food. UVB radiation converts precursors present in the skin into a form that the liver and kidneys convert into usable vitamin D. However, people living at high latitudes are often vitamin D deficient due to insufficient UVB radiation, especially in winter. Other factors also affect the production of the vitamin in the skin: the time of year and day, cloudiness and air pollution, or the use of sunscreen. Aging or darker skin can also slow down vitamin D synthesis. It is estimated that over 1 billion people worldwide suffer from vitamin D deficiency. This vitamin is essential for the proper functioning of the body. Its deficiency can contribute to various health problems, including immune and neurological disorders, memory and learning problems, and the development of schizophrenia or depression. Deficiency is also linked to a higher risk of developing cancer and dementia. There is also evidence that low vitamin D levels are associated with a higher risk of cardiovascular disease and other health problems. Studies have also shown that vitamin D deficiency is associated with increased severity of COVID-19 infection. Plants are generally a poor source of vitamin D, therefore. Most of it is obtained from animal products that contain precursors of vitamin D. These are products such as eggs, meat and dairy products. Vitamin D can also be supplemented. Tomatoes contain one of the building blocks of vitamin D3, provitamin D3, 7-dehydrocholesterol, 7-DHC, in their leaves, but at very low levels. Provitamin D3 does not normally accumulate in ripe tomato fruit. Professor Kathy Martin of the John Innes Center and her team used CRISPR-Cas9 gene editing to make changes to the genetic code of tomato plants. Eliminating a single gene caused provitamin D3 to accumulate in the fruit. According to the authors of the study, such a tomato provides about 20% recommended daily intake of vitamin D. Tomatoes and other plants in the nightshade family produce the precursor, provitamin D3, but then convert it into other compounds using enzymes encoded by two genes, 7DR1 and 7DR2. The researchers suspected that turning off one of these genes would cause provitamin D3 to accumulate in the plant. It fell on 7DR2, which helps in the synthesis of compounds used by plants to deal with stress caused by pests and microbes. Leaving 7DR1 alone, the study authors believe, resulted in the modified plants growing normally. When growing tomatoes, the leaves are usually waste. But those from edited plants can be used to make vitamin D3 supplements or for food fortification. The leaves of the edited plants contained up to 600 micrograms of provitamin D3 per gram of dry weight. The recommended daily intake of vitamin D is 10 ug for adults. We have shown that it is possible to enrich tomatoes with provitamin D3 using gene editing, which means that tomatoes can be developed as a plant-based, sustainable source of vitamin D3, said Professor Martin. 40% of Europeans are vitamin D deficient, as are 1 billion people worldwide. Not only are we addressing a huge health problem, but we are helping producers because the tomato leaves that are currently going to waste can be used to make supplements, she added. In previous research into the biochemical pathways of 7-DHC, scientists determined that a specific enzyme was responsible for converting provitamin D3 into other compounds, S17DR2. Then, using the CRISPR-Cas9 gene editing method, the researchers turned off the production of the enzyme so that 7-DHC accumulated in the tomato fruit. After this treatment, there was a significant increase in provitamin D3 levels in both the fruits and leaves of the edited plants. The researchers then tested whether the 7-DHC in the edited plants could be converted to vitamin D3 by exposure to UVB light. They tested it on leaves as well as sliced tomatoes, which they irradiated for 1 hour. It turned out to be very effective. The edited tomato, when exposed to UVB light, contained levels of vitamin D equivalent to 2 medium-sized eggs or 28 grams of tuna, both of which are recommended dietary sources of vitamin D. What's more, researchers say that vitamin D levels can be increased by prolonged exposure to UVB radiation, such as when drying in the sun. Blocking the enzyme in tomato had no effect on tomato growth, development or yield. Other closely related plants, such as eggplant, potato and pepper, use the same biochemical pathways, so the method can be applied to these crops as well. The COVID-19 pandemic has helped to highlight the issue of vitamin D deficiency and its impact on our immunity and overall health. The provitamin D3 enhanced tomatoes we have developed offer a much-needed plant-based source of vitamin D. This is great news for those following a vegetarian or vegan diet, and for the growing number of people around the world suffering from vitamin D deficiency, said Dr. Ji Li, co-author of the study. The modified tomatoes were grown exclusively in laboratory greenhouses. Field trials in the UK are set to begin next month. These tests will be crucial to verify that the plants can cope with real stress conditions. Acceptance of edited tomatoes by consumers may also be a challenge.